This is episode 124 of the Immunology Podcast, HIV Latency with Dr. Sharon Lewin. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Sharon Lewin from the Doherty Institute on the podcast to talk about her research on HIV. We also got our usual roundup of recent highlights in immunology news coming up. But first,
have you saved on a registration to Immunology 2026? Take advantage of early bird registration discounts today. Plus, refer to travel, hotel, and Boston area resources curated by the American Association of Immunologists. Join peers, colleagues, research, and industry scientists, potential collaborators and funders, exhibitors, and experts in the field from April 15th to 19th. AAI hopes to see you there. Register today at immunology2026.aai.org.
all right. So you got to go skiing recently.
Yeah, and you did not get to fly.
I did not. Nope. We shut down almost the whole country from a giant snowstorm followed by ice and ice and then cold.
Well, my condolences. I hope you guys get
I'm really hoping
quickly.
Like, we get a couple of days in above freezing a little bit, and so I'm hoping it all melts. It should, but it's a little rough.
All right. Okay. Enough of the weather.
Yeah.
Why don't you? Let's talk about the science. That's nice and warm because it's hot off the press.
It is. There you go. It's definitely hot off the press. All right. I'm going to talk to you about a paper that came out March 5th, which is in the future, in Cell.
Like in the long future.
I know, right? Sympathetic epithelial crosstalk governs tissue-resident memory T-cell immunosurveillance in the skin. First authors, Peng Zhang. Corresponding authors are Ting Zhu and Bing Zhang. All right. So sympathetic nerves, right? Adrenaline, flight or fight. They found, so there is a known effect where more CD8 tissue-resident memory cells, TRMs, are protective against cancer, right? Because they're going to see them there and kill them, the cancer cells. But they were trying to understand how that's regulated in the skin. And so they first looked at sympathetic nerves and tried to understand, okay, there's sympathetic nerves near these CD8 cells. Okay. On the skin. And then they're like, and so they showed that if they did a dread receptor, and that's those receptors that are designed to be exclusively activated by drugs, so you can inhibit the sympathetic system. In mice, it goes away. So if you basically blast them with a chemical that inhibits their sympathetic system and CD8 resident, the CD8 TRMs go bye-bye. So that was established, that's a thing that happens. And then they also did it through another chemical that wasn't, genetically derived. And then they're like, okay, well, let's knock out the main receptor, ADRB2, on all different types of immune cell populations, because that's clearly what's going to go on here. And then we'll see which immune cell population is driving this effect. And that did nothing. And they're like, okay, well, if it's not the immune cells directly that are being influenced here, right, by the signaling, then what is it? And so they went and found it was keratinocytes. And so they figured out, again, using genetic models, that it's the keratinocytes that really drive this. So you have these keratinocytes that respond to sympathetic stimulation or inhibition. If you stimulate it, the cells go away and you have more cancer, or you have more, eventually they show more melanoma, but they go, there's more of these cells. And if you inhibit sympathetic drive, you have more cells. Okay. So they're like, how? And So they started then dissecting what pathway could be used here. And so they eventually determined it was the chemokine receptor. Oh, and really quick, correction on the author, because they finally get here. There's two first authors, Peng Zhang and Juju Miao. So then the authors go on and pretty elegantly dissect the pathways out and identify the critical chemokine here that's driving this, which I'll get to, which is CXCL16, and discover that the keratinocytes secreted and pull in the resident memory cells. Because if you ablate the resident memory cells receptor, or you ablate the chemokine in the skin cells, it stops working. Now, the cool thing on these mice is they can use they can locally inject chemokine inhibitors or adrenergic chemicals or the special drug for the dread receptors into one side of the back subcutaneously versus the other with saline. And so you have a same mouse control in a lot of these studies because it's the skin, right? So they can look at local effects.
Cool.
And then they go and take this to a melanoma system and show that if you block melanoma, or if you put you use a melanoma system, the cell, sorry, take a step back. They also use an OVA system and show that the cells that move are clonally related, right? So there's not it's not driving proliferation, which was a question. It's not driving proliferation of the resident memory C cells. It's driving recruitment, which is how they got to it being a chemokine-based effect. And they showed that if they did some type of signal that causes clonal expansion in OVA plus CFA, you get more clones at the site, if that makes sense. So you have the skin cells pulling clones to the location. You're not having them reproduce based on less sympathetic drive. It's migration, not reproduction. And then they use a melanoma system, which, like in this system, immune cells suppress it. And if you have more resident memory cells there, it's known that you have less of this cancer. And guess what? When you do sympathetic inhibition, you get more, you get less cancer. If you turn on the system through acute stress or chemicals, like, good old adrenaline, but they're a little more sophisticated than that, you get, they use a chemical that hyperactivates it, you get more cancer growth. So basically, chilling out and not being stressed in your skin through inhibition of sympathetic systems pulls more resident memory CD8 cells there, which help immune surveil against cancer.
My. Again, another, it feels like almost holistic medicine or something. It's like, don't be stressed because that's going to affect your keratinocytes. Yeah, like 2 steps removed, like, you need your epithelial cells to be chill. So the T cells get recruited. So meditate. It's like, but I have cancer. It doesn't matter. Just meditate. I don't know. I mean, I find it very interesting. I think there is enough anecdotal evidence to suggest that not being terribly stressed is good for you. So I guess this having some insight on mechanisms by which this connection is explained is very, very interesting. So yeah.
Yep. Don't be stressed. It's going to cause you to lose your chill.
Yep. Very interesting. Man. Okay, so from my side today, I have a very B-cell centric selection today. And it's cool because I have B cells doing opposite things here under different circumstances. So I think it's a nice combination of papers I have today. So I want to start with B cells being bad, I guess, or B cells being problematic. So this paper is published in Science Immunology, I think 30th of January. And it's called B cells drive CD4 T cell immunosenescence and age-associated health decline. And first authors are Sad Khan and Mainak Chakraborty. And the senior authors are twin brothers, Sean and Daniel Winner from the University of Toronto. So I thought that was very funny. How would you like to have a lab together with your twin brother?
Creepy.
I think it's great. I think it's great. So, well, on to the paper itself. So, as the title already kind of gives away, there's a lot of interest and there's a lot to understand about the role of this inflammaging, quote unquote, a process in which with age comes this chronic low grade inflammation that increases as we get older. And that is linked to a lot of the pathology and age related complications of old age. And in particular, we know that it's, associated with the decreased vaccine efficacy, increased susceptibility to pathogens, increased uh, proportions of activated B cells, T cells, and kind of inflammatory mediators, and that's something that is kind of well known and well established. And so, in this paper, I don't know, I find it kind of cool that the concept behind this paper is pretty simple. What happens if we don't have B cells in terms of this inflammation? Because in the same way that T cells have suffer kind of clear changes in their repertoires in the sense of the proportion of naive T cells versus effector memory, central memory, there's an increase in effector subsets. B cells also change. And in humans, there is these, well, and there's the concept of a age-associated B cell that I think is better defined in mice and kind of being translated into humans. And these are B cells that are expressing CD11C. I think also some markers. So they're kind of a little bit more inflammatory. They are also expressing, so it's mostly there's a Tbet positive. They in the context of so they increase in amount in elderly mice. And also there's some populations in human that I think they're more associated with what is called a term that I don't think people use so much, which is called atypical B cells that are associated with human studies of chronic infection, in which you have, again, this CD11C positive, Tbet positive, highly T cell active, B cells are capable of activating T cells very strongly. And this kind of phenotype accumulates with age. And so in this paper, basically what they do is they have mice in which they have they are knocked out in B cells. So of course they can only do this because they have mice in pathogen free conditions. So they're not gonna die. Of course, if you don't have B cells in the real world, you're mostly gonna die. So sometimes it's when you look at the conclusions of this work, we mustn't forget that there's a trade-off. There's a reason why we have B cells, and it's not just to drive immunosenescence, but in order to to protect us from disease. But this, in a way, this paper shows that this protection comes at a cost. So what do? They have this B cell knockout cells and knockout mice, and they compare. They look at the mice as they age, and then subsequent experiment just shows their role in T cell and kind of inflammation in general. So they when they look into age, when they remove the B cells from these aged mice, they look around 2 years of age, so 24 months. That's quite a senior mouse. And they see basically a reduction in this inflammaging and in this aged immune system phenotype, and particularly on the changes that happen with age in the distribution of naive T cells versus exhausted and effector memory T cells and central memory T cells. So there's more of a conservation of naive T cells in both the sense of their phenotype, but also a reduction in the loss of clonality that is also very associated with age. You end up having expanded clones, you have less diversity on your T cell repertoires, and this is something that they see at least partially corrected by the absence of B cells, indicating that B cells are playing an important role in driving this clonal expansion / differentiation of T cells. And in normal mice, part of these B cells that are expanded, mice that do have B cells, are these ABCs, these H-associated B cells, and which is also, so in this case, they are defining them as CD21 negative and CD23 negative, CD11C positive, TVET positive B cells. And so that is probably the most, this result with the absence of B cells is the most, is the defining result of this paper, is the fact that by removing the B cells, you end up preventing a lot of the inflammation. And when they look at they stimulate the T cells from this B cell knockout mice, and with PMA ionomycin, they also see that they are producing less inflammatory cytokines, and associated with a more naive phenotype in general. And they're not proliferating so much ex vivo. So a lot of measurements they do, and they show that that if you are, they are kind of less differentiated, but also if you stimulate with CD3, CD28, you have the cells that activate very well, similarly to what naive cells do. So again, indicating that they have characteristics that are associated with a younger phenotype. And this translates in the mice, this translates in better health span and lifespan, and they have these frailty scores that are improved, and which kind of indicate the quality of the aging of the mice. And in general, these mice have better, less senescence-associated phenotypes. This SASP, the secretory phenotype, they have reduced fibrosis. So they basically, again, they seem to be spared from a lot of the hallmark signs of aging. And they see that they can, even if they are depleting B cells using anti-CD20 monoclonal antibodies, like our rituximab equivalent, they can also improve the abundance of naive cells, even if they deplete B cells in kind of middle-aged mice, 14 to 15 months old. And this actually helps restore or kind of recuperate some of these quote unquote kind of young T cell phenotypes, and improves also the kind of the overall health of the mice. So how is these what is connecting the B cells and the T cells and the aging? And they show that a key factor here is signaling through the insulin receptor in the B cells. And insulin receptor, so hyperinsulinemia, is also associated with age. And we know that activation of the activation of the insulin receptor it has a lot of also metabolic effects, and it also activates pathways like P13 kinase and AKT that drive mTOR activation, and these are all associated with inflammation and immune cell activation in general. And so what they see is that these B cells that are the C11C positive, TVET positive B cells that are associated with age, they are expressing the insulin receptor, and they're responding to this stimulation through the insulin receptor. They have increased activation of the mTOR pathway. And this seems that, so they conclude that the insulin signaling is, in a way, supporting the expansion of this particular cell subset. And these cells, because they have increased expression of MHC class II, which is dependent on insulin receptor signaling, which they show using flux mice, that this is kind of the connection between the age, the B cells, and the effects on the T cells, because they are being stimulated through increased amounts of MHC class II on these B cells. And this is kind of mechanistically how the B cells are inducing this activation and this aging of the T cell compartment directly. And that these T11C positive, Tbet positive B cells are important in connecting this compartments. And so I thought it was pretty cool because, again, there's a lot we don't understand about the role of this, especially the adaptive immune system, in both associated with age, but also causing the symptoms of aging, so to say. And of course, we're not going to get rid of our B cells because we need them to not die of infection. But I think it's very interesting to see whether we could, in a way, modulate the B cells that could actually have some meaningful effect on our immune system as a whole and how we age as a whole.
So it sounds like just by doing their job, they cause other cells to do their job, and that makes them get aged.
Yeah.
So
Yeah, in a way, yes.
just get tired from doing work.
Yeah. Yeah. Yeah, but I think that this B cell, so this particular C11C Tbet, I think they are, well, I'm not a B cell expert, so sometimes to me, they feel like they're a bit an overkill. Like you see them in the context of chronic diseases, and they're usually associated with negative effects of B cells. So it looks like they are a slightly undesired version of B cells.
Every cell has a main purpose for why it exists. Otherwise, we would have
Yeah.
long ago. So I bet you they're needed for infection and getting and coordinating inflammatory responses and inflammation, oxidative damage, and getting old. And so, sure, if you get rid of all inflammation, you can age in glory and then die of a pathogen before you get old.
I mean, that is in a way, for what I see, that's kind of the trade-off. You're stuck with this. It's much more important to keep you safe from a pathogen than to prevent you from aging. Once you have kids, who cares? Evolution doesn't.
Yeah, exactly. Like, are you going to make it or are you not going to make it? That is the question. Well, I have a story that definitely happens with aging. It is glorious. It is microbiome mediated, and it is wacky. So pretty much every one of my check boxes have been filled with this paper. So it is in Nature, coming out the 28th of January, which this time is in the past. Intestinal macrophages modulate synucleopathy along the gut-brain axis. First author is Sebastian de Schepper. Co-second authors, I think I'm doing this right, are Victorious Constanellos and James Conway. And co-last authors are Soyeon Hong and Tim Bartles. So in Parkinson's disease, there is that's called alpha-synuclein pathology, right? So that's where these alpha-synuclein proteins get aggregated. You get these Lewy bodies and other things, and then when they're in certain neurons, you no longer have motor control because it borks up your motor control of your body when these neurons go and die. That's basically Parkinson's disease, explained very, very briefly. So one of the interesting things is that constipation often occurs very early in Parkinson's disease, sometimes decades earlier, before motor symptoms. But constipation is a motor problem in and of itself, right? And there are T cells responsive to alpha-synuclein, AS, or alpha S, as I'm going to call it here. So the question is, does Parkinson's start in the gut and then spread to the brain? And because we all love efficiency these days in our busy, busy lives, is the answer apparently from this paper is yes. Yes, it does. So there's an enteric nervous system. And what they do is they create they use a mouse model, but they also can inject human proteins. So in this study, they use a mouse model that has this alpha-synuclein buildup. And then they also have the proteins themselves from mice and people they can inject. These bad proteins versus good protein, or they'll get bad neuronal tissue or good neuronal tissue and proteins from humans. And that's the basis of these models, right? So they'll either have a mouse model that develops more of these misfolded proteins, which leads to Parkinson's symptoms, or they will have injections into the gut of either human or mouse. They'll usually do a lot of mouse work and then show human too, right, to lock it in. So they identify that this really happens in the myenteric plexus more so than lamina propria, where this synuclein gets absorbed. When they do in just the baseline models, they see buildups there early on, or when you inject it, it'll build up there. They see a lot of macrophages in here doing their work, and so they're like, why is it that what are these macrophages doing? So what the macrophages do is they chew on all this naughty protein, because that's what they're supposed to do, right? And they establish that it goes in and it sits there and hangs out. And if you inject into into the enteric area, these proteins, you get this propagation from the enteric nervous system into the CNS over time of Parkinson's pathology, based on both symptoms of the mice and looking at the brains. And you usually see the tissue first, right? You can see the tissue damage before you have the symptoms. So how the hell is that happening? Well, they see more T cells, and more T cells that are clonally aligned to this protein misfolding go to the brain over time. They're like, okay, so something about the macrophage uptake makes more T cells trained against this protein to go to the brain. Okay, well, those T cells then go into the brain and destroy the brain cells that have the same proteins, because that protein is there, right? This alpha S protein is there, and that leads to the damage. So it's CD3 positive, CD3 T cells, not particularly specific. They can see the damage. They then try to figure out, well, how is this being conducted? And so they then look at the macrophages and try to understand the signaling there, and determine that mesenteric macrophages through TGF-1 beta are basically activating the T cells and telling them to go find other proteins in the body that have this bad naughty protein and go kill them. And so the macrophages are uptaking this bad naughty protein early from misfolding in the gut, accumulating it there, and then telling the T cells to go, hey, this is bad, go find this everywhere else. And so if you do macrophage depletion in the intestinal area, you get rid of the entire phenotype. And so the story of Parkinson's, which is very to me, this is a big deal, is that it starts in the gut, develops immune priming in the intestine, and then conducts along the enteric nervous system, or along the gut-brain axis, not the enteric nervous system, on the gut-brain axis. But these cells are near the enteric nervous system. This is the enteric plexus, right? The mesenteric and plexus, telling them to go up to the brain and go finish the job there. And so maybe if you start having atypical constipation, you could screen for this and then go on some type of therapy to suppress it so your brain doesn't get fried and you don't get actual Parkinson's, because the constipation is such a leading symptom. And there you go.
So I'm a bit confused about, in this case, so TGF beta
That's how the
is
macrophages tell the T cells to go have a party.
is there other cytokine? Because I always think of TGF beta as more of an anti-inflammatory cytokine. So I'm a bit, yeah, I wouldn't expect that.
It was a little weird.
Are these T cells that migrate, that are responding to these macrophages? What kind of phenotype do they have? Do you know
Oh,
if they
they
look?
looked at they just mostly looked at the total T cell compartment, but they were
Mm,
clonally interested in alpha synuclein. They were primed to target alpha synuclein. So they're not just
yeah,
any T cell, right? They're T cells that have been coordinated by the macrophages to have the same target.
yeah. I guess that's it's yeah. It's not surprising because we knew that there is this clear connection with the gut, and then this what you mentioned, the
Yeah.
um so I think, and it also makes sense given, the environment of the gut, that something would
And
get primed in there.
if you knock out TGF beta, the effect, the Parkinson effect goes away. The cells
Yeah,
don't go, they don't cause disease.
yeah.
Yeah.
That's very nice. That's very interesting. I wonder whether, yeah, I guess definitely could have a improve
Yeah.
screening, and I wonder whether something can be done, because if it's just starting, maybe still you're still in time to
Yeah.
prevent
And to be clear, these are the
brain.
muscularis macrophages. So not the lamina propria. It goes epithelial cell, then lamina propria,
Mm,
then muscularis. And the
okay,
muscularis is what touches the enteric nervous system, right? Because the enteric nervous tells the
right,
muscles to move, right? So it's those macrophages, the ME macrophages.
right. Yeah, they have to be there close to where the
Yep.
protein is to be found. Very interesting. All right. So why don't I close this session today with another B cell paper, as I promised, and this one is published in Immunity. B cell intrinsic IL-2 signaling regulates inflammation by promoting IL-10 expression in CD25 positive age-associated B cells. So again, we're going to the age-associated B cells, and in this case is mechanisms of kind of regulation of uh, more tolerance. And first author Juliette Gauthier, from corresponding author Céline Delalloy at the University of Rennes in France. And this is also really interesting. Another take on the role of these different subsets of B cells in inflammatory settings. And in this case, I think it's not so much about aging, although they are discussing this quote unquote age associated B cells. But it's more about their role in inflammatory environments and how these B cells respond to interferon gamma. They express Tbet. They are kind of um, again known for interacting with kind of inducing more an inflammatory environment. But in this case, this is not always the case. So that's, I think this kind of what confuses me a bit, as not being a big B cell immunologist. So it looks like there's still a lot of the nomenclature is still a bit confusing, because in this paper they are looking at a way for this ABCs to be more anti-inflammatory by producing IL-10. But let me start from the beginning. This paper, this kind of the premise behind this paper is IL-2. We all know good old IL-2. As a T cell immunologist, IL-2 is very much in my mind. And I think a lot about how IL-2 drives regulatory T cells, CD4 T cells, CD8 T cells, how important it is. It has been, was originally identified as a T cell proliferation factor. But B cells also express IL-2 receptor, including CD25, the high affinity IL-2 receptor. And B cells upregulate the expression of these receptors upon activation through their B cell receptor. And other stimulations, the CD40, a toll-like receptor engagement, things like that, activated B cells can respond to IL-2. So I guess the question is, what is IL-2 doing, and does it matter? Because in the end, yeah, it seems pretty clear that most of what IL-2 does is affecting the T cells, but let's see what are B cells doing. IL-2 drives the differentiation of plasma cells, and through mechanisms that include the repression of Bach2, which is a known kind of necessary step to differentiate cells that are, in the germinal center. So B cells, they get activated, they go to a germinal center, they interact with T cells, and they affinity modulate, and they change they do the isotype switch, and eventually they develop into plasma cells that are, producing a lot of antibodies. And these plasma cells have different transcription factors and different programming than germinal center B cells, and they're usually more kind of terminally differentiated. And what they see in this paper, kind of giving out the punchline, is that there is a coordination between different stimulations that include the B cell receptor, IL-5, which is known for kind of increasing the activation status of B cells in collaboration with B cell receptor stimulation. And that on top of this, IL-2 also affects the differentiation of these B cells. And what is interesting is that it promotes, by promoting Bach2 and preventing the loss of Bach2, promotes IL-10 expression in these B cells, and this ends up having a regulatory effect on the kind of the role of these B cells. So how does this work? What they do in this paper, they take IL-2, they knock out IL-2 receptor beta chain, and this results in B cells that are not capable of responding to IL-2, but also not IL-15. They show that it's very efficient, that there really is a loss of function. And although the B cells are mostly normal, there's a difference in how these B cells respond to stimulation when there's IL-2 present. So there you can see that B cells if they don't have the IL-2 receptor beta and they don't respond to IL-2, they differentiate differently. And basically, IL-2, the signaling the IL-2 dampens the plasma cell responses. So they have this T cell independent and dependent context. And basically, there's some intrinsic effect on the signaling of IL-2 that promotes, that restricts this differentiation into plasma cells and promotes regulatory functions. So if you have the knockout, you get more plasma cells. And basically what they show, they look at a lot of the details of the signaling, and they show basically that it is really IL-2 that is promoting the BCR dependent. There's some. Here's where it gets complicated, because there's some differences between the in vitro and the in vivo results. But just to keep it a bit simple, because otherwise it's too much. So when they take activated splenic B cells, so they take them from the spleen in vitro, the B cells that don't have IL-2 receptor have a more skewed response towards a pro-inflammatory program, and they have activation of STAT1, STAT2, 4, and TBED, which is this hallmarks of pro-inflammatory ABCH-associated B cells, and this that kind of moves them towards a more extrafollicular plasma cell differentiation. So there's this IL-2 is, in general, if you have signaling, you limit this differentiation. And they see that one of the hallmarks of this absence of this differentiation is an increased production of IL-10. And I think this is probably the most interesting result, because although they still see the differentiation of these ABCs, the most interesting thing is that they see an increase in the production of IL-10 when IL-2 is when these B cells are being stimulated with IL-2 and they have the receptor. So And they show that this the way that this works is through MAF, which is a transcription factor that induces IL-10 and is usually associated with anti-inflammatory responses. And the IL-2 MAF axis is what shapes a lot of this phenotype that they see in these ABCs, including the production of IL-10. And when they do so, they tested, they have MAF deficient B cells, and although they don't see differences in the phenotype of the B cells, they do see differences in the production of IL-10, which ultimately seems to be a major effect in the overall response. And in mice, if they have an EAE model, and they show again that if you have IL-2 receptor beta knockout mice, they have higher clinical scores. And this is associated with decreased IL-10 production that increases the EAE effect on the mice. So basically, I would say the conclusion of this paper is that B cells can sense IL-2. They're not indifferent to IL-2. And that under the right circumstances, if you combine IL-2 and B cell receptor, and also they look into interferon gamma in a situation where you have an inflammatory situation, interferon gamma, IL-5, B cell receptor stimulation, the addition of IL-2 and the sensing of IL-2 by these B cells results in them becoming an anti-inflammatory ABC rather than a pro-inflammatory one, and that this is mediated by IL-2 signaling.
Interesting that they sense it, but it's kind of a switch, but not completely.
Yeah, because the cells are still there. So I think this is what was a bit difficult for me to understand. It's their cells are still there. They're not, I don't see that they change a lot in the frequencies themselves, but it's what they do. But I think also this is difficult because the way they are defining these ABCs also is different in mice and in humans. So I was a bit kind of challenged by making sure I understood the subsets that we're talking about. I think the most important is IL-2 signaling together with interferon with with BCR signaling has this kind of unexpected effect of making IL-10 in these cells that are usually kind of associated with extrafollicular differentiation and inflammatory results and IgG production, things like that.
All right. Well, we're going to be talking about HIV here with Dr. Sharon Lewin at the University of Melbourne in just a minute. But before we get to that, scientific research is challenging, but isolating your cells shouldn't be. Proven in published research for over 20 years, EasySep allows you to easily and efficiently isolate the cells you need so you can focus on great science. Isolate a wide range of cell types from virtually any sample source in as little as 8 minutes. Request your free EasySep sample today at stemcell.com/easywin and share your experience for a chance to win prizes worth up to $3,000.
Now, for the second part of today's episode, we are welcoming Dr. Sharon Lewin. She is director of the Doherty Institute and professor at the University of Melbourne in Australia. And she has done very amazing work on HIV infection, and her lab tries to understand ways to cure or to improve the treatment of this disease, which is still a very important, very relevant topic nowadays. So, Professor Lewin, thank you so much for joining us today.
Great to be here. Thank you, Brenda.
Thank you for being here. All right. HIV research, right?
Yeah, that's right.
Okay. Why the heck is it so hard to cure?
Well, HIV is a pretty smart virus, and the reason why we can't cure HIV is because it has the capacity to integrate into the host genome. So once a cell is infected, HIV virus becomes part of the DNA and stays there for life. When you put someone on antiviral therapy, it blocks active viral replication, and it does that incredibly well. However, the virus, like many viruses, has two forms, a productive form and a latent form. And once it goes into latency, and it always goes into latency, it basically burrows into the DNA and sits there inside a resting memory CD4 T cell and stays there for life because those cells have a very long half-life.
So a lot of you, we've been working on HIV for a fairly long time, and it's very interesting to see how has our understanding of these limitations of therapy has grown in time, and how has the treatments that we can offer people also changed from that perspective. So where are we now? Maybe last 30 years since the peak of the epidemic, and now we've found solutions to keep people alive. We haven't been able to get rid of the virus, but how good are the options that we have now?
So the antiviral options we have now are absolutely fantastic. 70% of people globally living with HIV, and that's 41 million people, have accessed and are on these treatments. Most people are taking a single tablet a day, and that tablet contains either 2 or 3 antiviral drugs. You can't just take a single antiviral drug, but they're co-formulated into a single tablet. Or they're now beginning to take injectable antivirals so that you could have an injection every 2 months. That's less common, but a very exciting new area, because maybe one day we'll be able to give people an injection every 6 months or even every 12 months. There are clinical trials looking at that. For a person that goes on to this single tablet a day, the virus, which starts at very high levels in the body, you can measure it at in the range of 100,000 copies per mil of RNA, a million copies per mil of RNA. Within 3 to 4 weeks of going on to this treatment, the virus disappears to undetectable levels, or conventional laboratory assays that measure down to 20 copies per mil. Once someone is so-called undetectable, their viral load less than 20 copies per mil, we know it's still there, but it's undetectable by conventional assays. Their CD4 T cells can recover, largely because of thymus function, T cell proliferation, and you make new and better T cells. They're never quite normal. You always have persisting low-level immune activation, but your health does return to normal. Life expectancy returns to normal. And amazingly enough, when you're on antiviral therapy, you can't transmit the virus. That's a really important thing for people living with HIV. So nowadays we tell people, if you're on treatment, you don't need a condom if you have sex, because you cannot transmit the virus. And there's a big campaign called U equals U, undetectable equals untransmissible. So therefore you might ask, well, if antiviral therapy is so good, why do you actually even need a cure? What are we all fussing about? Well, antiviral therapy does come at a cost. Now, in my country, in Australia, where the government pays for the drugs that people living with HIV take, the average cost of antivirals is about $20,000 to $30,000 a year. That's when they're still on patent. But the rest of the world, low and middle income countries, can access treatment, generic drugs, and they're around $100 a year. Sorry, I think I said a day, $20,000 to $30,000 a year. But globally, in low and middle income countries, that's about $100 a year. So that's incredibly cheap. But $100 a year is still a lot of money in a low and middle income country, which might spend less than that on their total health budget. And to date, we've had amazing support from global institutions, primarily the US government, to make antivirals accessible. But whether that's sustainable forever, that we can cover that cost, we know is not likely, as we've just seen with disruptions with global health policy. So first of all, they come at a cost. Second, they come also with some personal costs. There's still a stigma and discrimination for people living with HIV. They have an infectious disease that's stigmatized and they have it for life. So many people with HIV really want a cure. And third, as I mentioned earlier, and given this is an immunology podcast, your immune system never quite returns to normal. So you still have low-level ongoing immune activation pretty much in everyone living with HIV. It's lower if you start treatment earlier, and in some rare people, it can return to normal. But that means that people with HIV are at a higher risk of immune-related comorbidities, which are, of course, we know heart disease is driven by immune activation, cancer, neurological disease, and these all occur at higher frequency in people with HIV. So taking that all together, the job's not done, as you said at the very beginning, Brenda. This is the holy grail. If we can work out how to cure HIV, which effectively means safely stop antiviral therapy, remain well, and not transmit the virus to other people, that would be an incredible advance, not only for people with HIV, but for our global funders. It's estimated that if we want to treat 95% of people with HIV, which is really the global goal, it costs on average $20 billion a year. So imagine if we could free up that amount of money for the healthcare system. So there's still a very important need for a cure.
So I'm going to put my hat on in two ways. One, 20 billion a year as an American doesn't sound too bad. Uh, but then secondly, won't if we have a cure, won't they just charge, 10 years worth of drug costs in the developed world for a cure? Because that's what pharma does, right?
Yeah, of course.
If it's 30,000 a year in therapy, it's a $300,000 cure. And now I got to go cure how many millions of people at 300,000 a pop?
Yeah. Good point. The health economics, not sure we need to get into that for an
Yeah.
immunology podcast. The health economics stack up if the cure intervention is costed at a reasonable level. Either way, one big difference with HIV is that if someone stops taking their treatment, and taking treatment for life is a really big deal. Imagine getting infected as a baby, which still happens. Most people get infected when they're sexually active, at their peak, which is usually in their 20s and 30s, and they're on treatment for 60 or 70 years. If you stop treatment, your virus rebounds back to its original levels in nearly everyone in 2 to 3 weeks, and you become infectious again. So there's a public health imperative to get as many people as we can on treatment. Where are we today? With enormous amounts of effort and funding, we're at 70%. We've never got, we're nowhere near 95% in many parts of the world. In my country, Australia, yes, we are. In the US, you're nowhere near that level of 95% in one of the richest countries in the world. So it isn't an easy feat to get everyone on treatment. And then get everyone in care for 60, 70 years and get everyone to be on treatment forever. So there's a health imperative, and that also factors into the economic burden, because once you go off treatment and you infect someone else, then that becomes another, $1 million addition to the healthcare costs. So there's been quite a lot of modelling done. The treatment of the intervention is important, but if it is because of the nature of HIV being an infectious disease, there is an economic benefit to this approach.
Okay, so let's go back to the immunology behind HIV and their study of this disease of AIDS. So one of the big issues is that you can get rid. You mentioned it. You can get rid of the circulating virus, but you're gonna have this latent virus, these reservoirs that are going to be mostly in CD4 T cells, or maybe not, and they're gonna become activated as soon as you. So they're gonna be obvious as soon as you. Pause, or as soon as you remove the treatment, or are there what are the kinds of triggers that result in this reservoir activation? And I know that some of your research has gone into understanding these reservoirs, and what is the angle? Can we get rid of them somehow?
Yeah. So the reservoir is predominantly CD4 T cells. About 99% of the virus in people on treatment is sitting in CD4 T cells, a small percentage also in monocyte macrophages. For a long time, we thought that the virus was sitting just in resting memory T cells, but actually it's sitting in all T cell subsets, naive, memory, effector, in activated. We always have a small number of activated T cells circulating, even when you're fit and healthy, in activated, in resting T cells. And over the years, we've learnt that the virus has got different levels of latency. So when I say latency, what I mean is the virus is integrated in the host genome and transcriptionally silent. As soon as that T cell is activated, because it is seeing its target antigen, as all memory cells do, they see something foreign, is suddenly activate, proliferate, and then revert to become effector cells and revert back to becoming central memory cells. The virus also gets triggered because activation of the T cell also activates the virus. But what we've learnt, and actually my work, but going right back to some of the earliest work I did in the mid-90s during my postdoctoral fellowship at Rockefeller, I was then working with David Ho, who was Time Man of the Year actually in 1995 for discovering why combination antiviral therapy is so effective. And he did the first studies together with Marty Markowitz of effective antivirals. We developed what was then very sexy assays, cell-associated RNA using real-time PCR, which was a big deal at the time. And what we found was that real-time PCR, which is of course now very basic, using sensitive assays such as real-time PCR, you could find HIV RNA always in people on antiviral therapy. In other words, the cells were not truly transcriptionally silent all the time. It gets a little bit more complex than that because HIV has quite a few steps in its viral life cycle transcription, like many, any gene and any in any RNA virus. Once it's integrated in the host genome, you get transcription initiation, elongation, completion, splicing, protein production, and then release of virions for the cell. And most of the virus that's sitting there is either integrated and silent or integrated and not and just undergoes transcription initiation, because a resting CD4 T cell is not the right environment for the virus to take off. It needs a lot of transcription factors, NF-kappa B, PTFB, a whole lot of other transcription factors present in an activated cell to complete its transcription. But think of it that you've got these viruses ready to go, and as soon as that cell's activated, bang, it's off. And what we see, and this is actually an area that's not really well understood, because if this was stochastic and you just were sitting ready to go in a antigen experienced T cell and it sees its right antigen and it goes off, you'd expect that the time to viral rebound after stopping antiretroviral therapy would be variable because it's stochastic. It depends on that T cell. And remember, only one in a million T cells carries a replication competent virus in someone on antiviral therapy. But actually, it's not stochastic. When you stop antiviral therapy, pretty much 95% of people rebound in 2 to 3 weeks. And that says to me that there's always some virus ready to go, ready to pop out of a cell. But while you're on antiviral therapy, you don't see it because it never can go on to infect new cells. So what my lab's done over many years is develop better assays to develop these low levels of RNA, understand where those cells are sitting in the body, because they're not all circulating. Most of these cells are sitting in tissue, in gut and in lymphoid tissue. And my own interest has been how to activate that virus and allow it to be visible to immune-mediated clearance while you're on treatment. So all those viruses that are sitting there ready to go, how do you get them to complete transcription so you can kill them while you're on antiviral therapy? And how do you do that without actually activating the T cell? Because
Right.
you can't use broad spell. You can't use IL-2 has been tried, and it's toxic. You can't use OKT3 or something like that to activate every T cell. So that's been a really big interest of mine, measuring and understanding RNA transcription, and then how do you get that going while someone's on antiviral therapy without activating the T cell. And we've tried 1,000,001 different ways, and some of those, most of those approaches have been not HIV specific, like histone deacetylase inhibitors. Because if you alter the epigenetic environment, you can fire off the virus. But none of those approaches have been HIV specific. I've led clinical trials looking at them in vivo, not that effective, get a little bit of RNA, but no T cell death. But over the last few years, we've got some really exciting work using HIV specific approaches and mRNA, and I can get into that later.
So, what is going to work potentially for a cure, right? We've looked at vaccination. That's been a real bear. I think we've all been hoping for that, and that's just been rough. What do we think is going to work?
Yeah. Lots of things being tested, lots of things looking promising. And the principles of trying to find a cure we haven't talked much about the immune response, so maybe we should get to that in a minute. But the principles are target the virus or target the immune system, reduce the pool of infected cells to as small as you can get it or to zero. Some of the approach that I use by activating the virus or using pro-apoptotic drugs, such as venetoclax, because these latently infected cells express high levels of survival proteins such as BCL2. So there's been a lot of work on inhibiting pro-survival proteins in cancer, such as BCL2 antagonists, a Melbourne-based discovery from WeHi, now licensed for the treatment of chronic lymphocytic leukemia. For example, pro-apoptotic drugs will shrink the reservoir. We've got a clinical trial underway looking at that. The flip side is to boost immune control, whether it's T cell mediated, NK cell mediated, or antibody mediated. And as you rightly said, obvious way is therapeutic vaccination, but that now hasn't really worked. There's been one or two studies that look marginal at best, but it's really hard to re-educate those existing HIV-specific T cells or generate new functional HIV-specific T cells. It may be possible, but vaccination hasn't done it yet. The areas that look promising to do that are using different forms of immunotherapy. So one form that I've been very interested in is anti-PD-1. So your listeners will know lots about anti-PD-1. In fact, PD-1 was identified as an immune exhaustion marker very early on, initially, of course, in lymphocytic cytomegalovirus, in LCMV, but very shortly after identified in chronic viral infections as being elevated in your viral-specific T cells, particularly HIV, but then shown in hepatitis B, hepatitis C, and other human infections, as well, of course, as in cancer. So in clinical trials of anti-PD-1, and there've been there's now emerging a few of these studies. When anti-PD-1's given, we've done a lot. My own lab's done a lot of work on when you give people with HIV with cancer anti-PD-1. We just did opportunistic observational studies. But when you give people with HIV on antiretroviral therapy, anti-PD-1, and stop treatment at the same time, so you're giving antigen exposure as well as an intervention to reverse T cell exhaustion, there is some initial promising results that a subset, about 20% of participants, can control virus after receiving a short course of low-dose anti-PD-1. So we know that, and actually in monkey models, other work we've done together with Afa Makoyu and Lewis Picker at OHSU shows, again, if you give the anti-PD-1 at the time of stopping antiviral therapy, not while on antiviral therapy where there's no antigen or very little antigen around, you can re-educate the existing T cells to no longer be exhausted and functional, and you can lower viral load. So there's quite a bit of work going on anti-PD-1. We're currently doing a clinical trial here and soon to open in Singapore using low-dose anti-PD-1. Of course, the problem with PD-1 is immune-related adverse events, which are acceptable in someone that has cancer, but not in people with HIV. So that's one form of immunotherapy. Another form that that's had several clinical trials now, about 5 that have shown some efficacy, is to give antibodies. And the antibodies that have been tested are broadly neutralizing antibodies. I think people will understand what that is, but in brief, some people with HIV make very good endogenous antibodies that can neutralize a wide range of variants. And if you isolate the B cells from those people and clone the right genes, and you can manufacture broadly neutralizing antibodies. There's been a large number of broadly neutralizing antibodies described, several that have reached clinical trial, and 2 that have been licensed to Gilead. And when you give those broadly neutralizing antibodies at the time of stopping antiviral therapy, in some participants, you can also induce control. The virus is still there, but it's controlled at low levels. So they look quite interesting, and now people are using combination immunotherapy. Recent paper in Nature from a group at UCSF showing that if you give a combination of immunotherapy, vaccine, broadly neutralizing antibodies, TL1 agonists, they could induce control, not to the level that we need, but it's that you can shift the dial of immune dysfunction. And one last area that I think is really exciting is gene therapy. And there's a whole lot of ways gene therapy has been tested to actually CRISPR-Cas9 to target the virus. Clinical trials not yet shown to be effective. Insertional gene therapy to insert a gene of a BNAB into the host to produce antibodies ongoing. That's been done successfully in adults, but really promising in infant macaques because the gene is persistently expressed and basically the virus can't come back because the person's making their own endogenous antibodies.
Right.
And gene therapy like CAR T cells, and also this, I mean, the work we're doing with mRNAs, it's essentially ultimately going to be a form of gene therapy. So there's lots of really exciting stuff that's being tested.
Yeah. Or you can just get a bone marrow transplant with a CCR5 delta.
Yeah, I mean,
That's a bit
of
tougher,
course,
right?
yeah, that is worth mentioning because it's proof of concept that that the reservoir can be wiped out.
Yeah.
And actually in those cases, now 9 cases reported around the world, not all with interestingly requiring a CCR5 negative donor. 2 of the 9 have not had that. One had a wild type donor, one heterozygous for CCR5. You truly wipe out your reservoir, and that's a mix of graft versus host disease, the conditioning treatment, and having a new marrow with cells that you can't infect. So that's inspired many new directions of treatment.
For sure. So it's great to see that there's so many different avenues for treating this very elusive pathogen. I think that's all what we have time for now. But we just want to ask you one quick last question. So we like to ask our guests something to get to know them a little bit better. And our favorite question to ask is, if you were not, a world-renowned HIV scientist, what do you think you would be doing with yourself? What would have been your alternative universe?
Yeah, well, I had always wanted to be an astronaut, actually.
Oh,
I
that's
was
great.
really interested in physical science, loved physics and maths at school, kind of went into medicine, not terribly interested in medicine, to be honest, but grew to love it. In Australia, you go straight into your chosen field, or you did in my day, you don't do an arts degree first. Yeah, I would have been an astronaut, and I still look or an engineer. When I look at these massive, big bridges, I think, wow, I would have loved to have built that. So I think astronaut or engineer.
Sounds great. That's a great answer.
Maybe you'll send an experiment to space one day, the thing.
Might affect the viral reservoir. You never know.
Indeed. All right. So it was great to have you. We had with us Sharon Lewin from the Peter Doherty Institute in Melbourne, Australia. Thank you so much for joining. It was a great conversation.
Thanks very much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get the show notes, including episode summaries and links to all of the interview and Roundup Papers. If you enjoyed the episode, feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at Immunopodcast, Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.